Hello, welcome to our YouTube channel. Today we're going to be doing our Unit 6 project on genetics by Maria Milan and Yamila Mendez. We are going to be focusing on Down syndrome and Ewing sarcoma. Down syndrome is a genetic disease when chromosome 21 has an extra chromosome. In a case of Down syndrome, the sex cells go through meiosis 1 and comes out perfectly fine as normal cell division happens. So entering meiosis 2, prophase 2, the spindle fibers form and begin to move the chromosomes towards the midline of the dividing cell. So in metaphase 2, the chromosomes move to the midline of the dividing cell facing opposite poles of the dividing cell. Anaphase 2 is the source of Down syndrome. Usually in anaphase 2, the chromatids separate and move towards the opposite poles of the cell. However, this does not happen when Down syndrome occurs. What happens instead is the chromosomes don't separate, which is called non-disjunction, meaning that the chromosomes don't split up and stay together. Then the cells go through the next step, which is telophase 2. In telophase 2, a nuclear membrane forms around the chromosomes in each of the 4 new cells. The cells have reached the end of meiosis 2. Here we are left with 4 cells, each having 2 except one that has 3 chromosomes in it. This is chromosome 21, where it has 3 chromosomes instead of 2. Here at first we have a normal karyotype, but then we add another chromosome to chromosome 21, making it a Down syndrome karyotype, which is 21, the one we circled red. So here we have a pedigree chart of Down syndrome. So we have 2 parents who are unaffected, and then we have their 4 offspring. 2 of them will be affected by Down syndrome. However, Down syndrome isn't like X recessive or any of that, so it could happen to anyone. So some symptoms for Down syndrome include developmental delays, characteristic facial features, short stature, heart defects, and vision issues. However, this varies from person to person. Who is at risk? Anyone with family history of Down syndrome is at risk. However, also babies whose mother are older than 35 are at higher risk to getting Down syndrome. Sadly, today there's still no cure for Down syndrome, but there is therapy that helps with motor skills, such as speech, vision, and all that. They also use medical treatment that helps with the side effects. They use a drug called paracetamol that helps the mind focus and the brain to understand and learn.
The cancer we're going to be focusing on today is Ewing sarcoma. An underlying cause is a translocation between chromosomes 11 and 22, which fuses the Ewing sarcoma gene of chromosome 22 to the FLI1 gene of chromosome 11. There are 4 stages of the cell cycle: the G1 phase, S phase, G2 phase, and the mitosis phase. When Ewing sarcoma comes into play, it controls the proliferation by deregulating the G1 phase cell cycle regulator, which basically means that not all cell lines undergo growth arrest or change in growth rate with changes in Ewing sarcoma expression. The Ewing sarcoma protein results in cell cycle arrest in the mitotic phase, mainly due to an increase in the time from nuclear envelope breakdown to metaphase, resulting in a high percentage of unaligned chromosomes and multipolar spindles. It regulates the formation of mitotic spindle. The mitotic spindle is a group of spindle fibers that divide chromosomes during mitosis. In order for a cell to divide, they must be in contact with the solid surface. That's anchorage dependence. Density-dependent inhibition means that if a cell is crowded, it will not divide. Cancer ignores this rule and keeps dividing uncontrollably. A cause of density-dependent inhibition is lack of growth factors. Some growth factors are oncostatin M, which is a protein that in humans is encoded by the OSM gene, basic fibroblast, which is a growth factor and signaling protein encoded by the FGF2 gene, and insulin-like, which are proteins with high sequence similarity to insulin, platelet-derived, which is proteins that regulate cell growth and division. Here we have a group of cells and the red spot is the carcinogenic initiator. Below that we have the unrepaired DNA damage. This is the initiation stage. Here we have a group of cells with a tumor promoter. Then it leads to clonal expansion. The acquired mutation is from the initiation stage. That can turn into benign tumor formation. That is the promotion stage. Now we can see that if the benign tumor formation is left untreated, it can turn into a malignant tumor transformation. This stage is called progression. Now we're going to go over the differences between benign and malignant. Benign does not spread and it does not grow back after it's removed. Malignant can invade other tissues surrounding it. There's a chance it may grow back after it's removed, and it can break off and travel through the bloodstream. Now we're going to go into some treatments. To shrink the tumor and prevent new ones from forming, chemotherapy will be taking place. After the tumor is contained enough, our next option is surgery. But if the tumor isn't contained to destroy cancer cells, radiation is our next step. Lastly, we're going to go into survival rates. About 70% of children with non-metastatic Ewing sarcoma survive. The survival rate for children diagnosed after their disease has spread is less than 24%. As a child ages, the survival rate decreases. Teens aged 15 to 19 years old have a lower survival rate, roughly 56%.